All right, we can get started now. As we begin, I request everyone to kindly mute their mics to ensure there are no background noise during the session. To set the tone with wisdom from our ancient text, I would like to share a Tirukural. கற்க கசடர கற்பவை கற்றபின் நிற்க அதற்குத்தக. Learn thoroughly whatever you learn and let your conduct be worthy of that learning. Vanakkam and Namaste. I would like to extend my warm welcome to everyone present here and I wish you all a pleasant and productive morning ahead. Respected faculty members, beloved alumni, enthusiastic students, and our cherished virtual participants, it's truly an honor to have you all with us today. It gives me immense pleasure to welcome you all to the fourth lecture of the Physiology Alumni Lecture Series, PALS 4. As we celebrate the silver jubilee of our MD Physiology program here at Sri Ramachandra Medical College and Research Institute, we take this moment to reflect the remarkable journey of a department that has grown not only in numbers but in its impact, influence, and inspiration. Since 2000, we have proudly mentored 58 MG graduates, each one a spark contributing to the ever-evolving flame of physiological science. And with 10 promising minds currently in training, that torch continues to shine brightly into the future. PALS isn't just a regular lecture series. It's a celebration of continuity where the wisdom of experience meets the curiosity of emerging minds. A bridge across generations, a forum where textbook meets testimony. There is greatness in acquiring knowledge. There is glory in sharing knowledge. So now, to take us through her inspiring academic journey, I now invite Dr. Abhirami Om Prakash ma'am, Professor of Physiology, to formally introduce our speaker. Abhi ma'am, the virtual stage is yours.
Good morning. I'm extremely pleased to introduce our guest speaker of the day, Dr. Magna Manjareeka. She is currently working as Professor of Physiology in Kalinga Institute of Medical Sciences, KIIT University, Bhubaneswar, Odisha. She has completed her undergraduation from Medical College, Tamil University, and she completed her post-graduation MD Physiology from esteemed Medical College, SRNRI, Sri in Chennai. She is an esteemed professor having teaching experience of 14 years and an avid researcher who has guided and guiding many MD postgraduates and PhD scholars for more than 14 years. She is a core member of Medical Education Unit. She serves as a resource person for faculty development programs and medical education training programs in her institute. Her areas of interest include mental health, adolescent health, electrophysiology, and heart rate variability. On the topic, the brain's blueprint, decoding the physiology of learning and memory. Over to you, Dr. Magna.
Thank you very much, Dr. Abhirami. Before I start my topic, thank you. I want to thank Priscilla ma'am very much. I am what I am today because of Priscilla ma'am. Thank you very much, ma'am. and nice to see Muthu, Pratibha, and Abhirami. Muthu, Pratibha, heading a department in Ramchandra. Actually, I am very much connected to Ramchandra. In fact, when I completed my MD, I really wanted to join Ramchandra also. But as all many of you know that I'm a native of Odisha, so I got an opportunity over here. So I came back over here, and I am working in a equally efficient institute like Ramchandra at KIMS and KIT University. Ramchandra had NIRF ranking in 2024-16, and I think we had in 21. So it's almost a very efficient institute. And whatever knowledge I have gained staying at Ramchandra from Prisla ma'am, Padma ma'am, Subhashini ma'am, I try to convey all those things, and I have learned also many things from my seniors. So Ramchandra is actually a sweet memory for me. So I really feel connected. I really feel connected. Thank you, Pratibha, for giving me this chance to be connected with you people again.
Our pleasure totally, Magna, to have you with us. It's a joy to see you, and you haven't changed a bit. And I think many of us are here together because of Priscilla ma'am. Ma'am, it's good to see you also. And thank you, Abhi, for getting things started. So over to you, Magna. Have the stage is all yours, and have a great time with us.
Thank you, thank you. Ma'am, with your permission, can I start? Prisla ma'am?
Yes, Magna, nice to meet you. Thank you.
Thank you,
Very
ma'am.
happy to see you. Okay.
Thank you, ma'am.
Waiting to hear from you.
Is my PPT seen?
Yes, yes, Magna.
Okay. So, good morning, everyone. Today's discussion topic is the brain's blueprint, decoding the physiology of learning and memory. As I've already said, and Abhi has elaborately told us, I'm a professor working in Kalinga Institute of Medical Sciences, KIT University. So, objective of this session would be all the participants should know a little bit about the history, the classification of learning, the physiological basis of learning, the types of memory, mechanism involved in the memory, and how learning and memory are interlinked, and some aspects of applied. So, what we exactly understand by learning? So, when we understand what is learning, suddenly it comes the present definition, or the layman definition tells us that it is acquisition of some information through studying, through practice or experience, which can bring about some certain change in the behavior. Now, in history, if we see, this started Skinner, B.S. Skinner was the first one who worked upon this aspect, and he defined learning as a progressive behavior adaptation. He didn't say it is a change, he said that it is a behavior adaptation. Gradually, when we see ligands called, they said that there is transformation of experience and practice, which leads to gain in knowledge and information. Then in 1988, the first step of learning by Vandenbaan and Hawkins, they said that through experience and practice, there is certain change in the behavioral pattern. And from there, we can see in 1990 ways, then Atkinson and Woolfolk, all these people, they also said that there is, they concluded that there is change in behavior because of acquiring some amount of knowledge, either through experience or by studying or by practicing, whatever. But if we see, the father of modern learning theory has been given to Ivan Pavlov. He was a basically Russian Soviet experimenter. He was a physiologist, and he had got Nobel Prize in 1904 for medicine and physiology because of his extensive GI physiology uh, experiments, where he proved. We all know that he had more contribution for the classical conditioning and operant conditioning elaboration. So he was a Soviet experimenter, and he was born in 1849, and he expired in 1946. Now coming to what is memory. So when we say memory, memory is defined as a process involved in acquisition of knowledge, storage, and retrieval of that knowledge or information. So if we see, this is the present definition of memory, where we can see that acquisition of learning, acquisition of information is a component of memory. So if and this so if we mutually, learning and memory, they cannot be excluded. They are mutually inclusive because learning is a part of memory. Now, what happened if you come to the history? Memory has been studied a long back. Plato and Aristotle, they said that the heart is the site of learning or memory. And gradually, the next era, the Greek and Romance, they said that they framed for memorizing things, they framed mnemonics, and they named it as the recall theory. But Hermann Ebbinghaus, who is the father of memory in psychology, in 19th century actually did scientific research and to prove that how memory is working, and he was the first one to have a scientific approach to the memory research. Now, gradually, Carl Lashley and many people contributed to this. And Hermann Ebbinghaus was actually a psychologist, German psychologist. And by 20th century, we had little bit of distinction between what is memory, what is the classification of memory, and such what the theories which are existing at the primary level existed by the late 20th century. Now, coming to learning and memory, as I said, they are two sides of a coin. So, learning is no way effective if we don't, memory does not exist, or memory is not possible if learning is not there. Learning is acquisition of the information, and memory is retention, storage, and retrieval of that information. So, both of them are interlinked. So, it cannot be a separable thing. Now, coming to the classification of learning, we have two types of learning. One is non-associative learning. The other one is associative learning. When we say it is non-associative learning, the animal or individual learns from a single stimulus. So examples are habituation and sensitization, which we'll see in details. Now, what is associative learning? Where the organism or the individual learns by associating two stimulus or stimulus to a behavior. So they are classical condition. The examples are the classical condition reflex and the operant condition reflex. Now, Kat Thompson, another psychologist, he divided learning into one third category where there is non-associative learning and associative learning. Along with that, he said that there is existence of observational learning, or you can say social learning theory. And we will go also to that. And in the observational theory, what is happening? By learning occurs through just imitating another individual, seeing what the other, or observing what the other individual is doing. Now, coming to the non-associative learning. Non-associative learning, as we said, it is due to repeated stimulus from a single stimulus. So here what happens, there are two types of examples of non-associative learning. One is habituation and the other one is sensitization. So we'll take two examples, then only we'll go into understanding actually what is habituation and sensitization. Many of us know habituation means whenever there is a repeated exposure to a single stimulus, gradually that the response is not elicited no more. So, like if somebody is staying very near to the airport, and the person is the sound of the noise of the planes or flights moving around that does not elicit any response. It does not annoy the individual who is staying near it, because the person has got habituated to it. But a new person who comes over there, the person says that how can you tolerate such type of annoyance? So the person who stays over there, because of the repeated noise, is avoiding the stimulus. He thinks that this ignores the stimulus and is eliciting no more response, which is he is having no more irritation or annoyance towards the thing. Now, what is sensitization? Sensitization means if there is a repetitive single stimulus, noxious or a pleasant stimulus, which is repeatedly given, what happens? There is an increase in response. So here in habituation, there was a decrease in response, and here this type of stimulus is causing a decrease in stimulus. So one short-term sensitization example we can give, where suppose we have seen a horror movie, and uh, after seeing the horror movie and we are going to fetch some water, and one of our friends just says who, then we get immediately scared. But after certain days, if that who comes, we don't get scared. So the horror movie basically sensitized the simple woo into a larger or a greater response. Now, to understand the different experimental basis which in learning and memory, actually the sea sloth, or you can say the Aplysia californica, was the organism which was used because it had simple nervous system. The neurons were very large, and it could be detailedly absorbed, and the researchers can also manipulate the activities to see the change in the responses. And it was a very effective organism for experimenting on all sorts of learning. And the main and important point was evolutionary conservation. They saw that Aplysia nervous system has certain amount of nervous tissue, the responses which were the cellular and the molecular mechanisms of learning and memory, which was seen in the sloth, were almost similar in the vertebrates. So that's why these experiments which were done in the Aplysia are corroborating with the findings with the vertebrates. Now we will see experimentally how the habituation and sensitization works. So this diagram is showing sea sloth. This is the siphon of the sea sloth, and we have the gill of the sea sloth. And naturally, if the siphon is touched, there is the gill withdraws. The gill withdraws. This is a normal reflex occurring in the so what happens when we the work is, how is the work? There is a sensory neuron which comes, and it releases the neurotransmitter to in the motor neuron, and it stimulates the motor neuron, and the motor neuron causes through the neurotransmitter release, it causes contraction of the gill. So we see the contraction of the gill. So this is the normal reflex which is there in the Aplysia. What happens if we give repeated benign stimulus to the siphon of the Aplysia? Basically, what happens? We see that presynaptically in the sensory neuron, the neurotransmitter release is decreased. Thus, in the motor neuron also at the neuromuscular junction, the neurotransmitter release is decreased. So there is no more gill withdrawal seen to a simple stimulus. So here, what has happened? Because of decreased the repeated benign stimulus has decreased presynaptically neurotransmitter, leading to less acetylcholine secreted at the neuromuscular junction and less response. So this is the process of habituation, where there is decrease in neurotransmitter release presynaptically, and basically that presynaptic release is because of it has been seen that there is decrease calcium influx presynaptically, thus the release of vesicles into the synapse is decreased. Now coming to the sensitization, what happens if there is any noxious or unpleasant or pleasant response? This is again a aplysia where this is the siphon, and you can see what happens if we keep on giving uh, 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 this is the normal reflex what is happening. But what happens if you are giving stimulating the noxious or unpleasant stimulus repeatedly? There occurs a presynaptic facilitation of the motor neuron. Thus, the gill will contract in a faster way or enhanced way. So basically, any obnoxious or unpleasant stimulus is when repeatedly given, the gill contraction is more. But after that, if a small touch stimulus is given, that also causes a abnormal or enhanced gill response, because gill withdrawal response, because of the facilitatory neuron, which is causing the release of excessive neurotransmitter, and that leads to an enhanced response. Now, if we see what is happening over here, is this diagram here. The facilitatory neuron which is present is releasing the neurotransmitter, that is the serotonin, and serotonin acts on the sensory nerve, on the sensory or the presynaptic neuron, and causes increase in neurotransmitter release by three mechanisms. So the serotonin which is released over here binds to two types of G protein receptors. One is GO and GS. In the GS receptor, when the G is binds to the GS receptor, it stimulates the adenine cyclase to produce more amount of cyclic AMP. Now the cyclic AMP activates the phosphokinase A, which is again cyclic AMP dependent. Without increase in cyclic AMP, this phosphokinase, active part of phosphokinase A, cannot be activated. Now this active phosphokinase A, what does it do? It inhibits the potassium channels. Once it inhibits the it inhibits the potassium channels, so potassium efflux is less, so action potential is prolonged in the thing, and when there is increased action, when there is the action potential is prolonged, the neurotransmitter release is for a more time, or you can say the influx of calcium is more, thus neurotransmitter releases. The second mechanism is the cyclic dependent pKa directly causes the neurotransmitter to be released at the membrane site, and the third one is it causes the long lasting calcium channels to open up, again causing the prolonged or causing influx of calcium, which again will help in release of these vesicle or neurotransmitter from the vesicle. The second receptor is the G protein receptor, and it activates or it works through the phospholipase C system, which again acts on the membranous diacylglycerol to form active phosphokinase C, and that phosphokinase C helps in release of more neurotransmitters through opening of the long-lasting calcium channels, thus increasing the influx of calcium, and also it directly stimulates the vesicular release at the synaptic sites. So ultimately, we see that the synaptic in the sensitization, the presynaptic or the facilitatory neuron releasing the serotonin is causing the presynaptic release of increased neurotransmitter into the synapse. Now, there is something known as long-term sensitization, where apart from all the activities what we saw is happening, but along with that, the activated phosphokinase A is translocated and goes to the nucleus, and there it phosphorylates the TREB1, which is a transcription factor. And once the transcription factor TREB is stimulated, it causes the response element, the nuclear response element to be stimulated, and results in the new effector protein production, and that will again enhance in a long-term way to increase the synaptic release of neurotransmitter. Now, if we see, to summarize the non-associative learning, we see that in habituation, the response is usually decreased to the whatever was the innate response, and usually it is a benign stimulus. And in sensitization, the response is always enhanced or facilitated after repeated exposure. And the cellular mechanism involved in habituation is presynaptic inactivation of calcium channel, leading to decreased neurotransmitter release. And in sensitization, again, release of neurotransmitter 5-HT neurotransmitter, which facilitates the presynaptic release of neurotransmitter at the sensory and motor neuron. Now, here, because of the benign stimulus, eventually the individual ignores the stimulus and there is no response. But here, let it be a benign one or a pleasant one or an unpleasant one, there is a greater response. Now, the examples, as I have already given you, in the noisy surrounding, initially though it bothers us, but gradually it does not affect us. But and for sensitization, after a painful stimulus, even a mild touch can incite pain. Now, there are two terms known as dishabituation and desensitization. So, what is dishabituation? Dishabituation is if this, we'll take the example of this case. If an individual who is staying in the near an airport is now habituated to avoid the noisy surrounding of the flights, still, if the person hears something apart, something different than the noise which he regularly or she regularly hears, then the habituation is no more seen. That becomes an annoyance. So any other noxious or a new stimulus, apart from this one or apart from the one which we are habituated to it, can elicit a response. And you can say that this habituation actually is one type of sensitization. So the new stimulus causes sensitization because of the dishabituation. Now, what is desensitization? If the distinct stimuli if the distinct stimuli is not given for a longer period, let's say there is no repeated one. The example what we saw is in the when we see a horror movie and our friend is a small who of our friend is making us scared, but after some times that sensitization, because that horror movie is no more in our mind, then small any other small mild stimulus will not make us. So we can say that the difficult or the stimulus which was sensitizing us to for facilitated response is when it is no longer there, then we are again desensitized. Now coming to associative learning, there are two types of associative learning. One is a classical and the other one is a operant conditioning, or classical conditioning and operant conditioning. So here, as we already know, either we see the association of two stimulus or effect of two stimulus, and here we see the association of a voluntary behavior with the consequence. So here in the classical conditioning, there is a neutral stimulus. When it is associated with naturally occurring stimulus, the neutral response also elicits some effect, and so there is pairing of both the stimuli. And on this actually classical conditioning, Pavlov worked upon it, and we will see in details what has happened, how it is done, or what is the mechanism. Now, in operant conditioning, the behavior, the voluntary behavior either is strengthened or weakened depending upon the consequence. If it is a good one, then it may be strengthened. If it is a bad one, then it may be weakened. So, and the operant conditioning was elaborately studied by B.F. Skinner, by the Skinner's rat Skinner box. Now, as I said, the Pavlov studied by the unconditional and conditional stimulus through a dog, and to see the effect of association of two stimuli. And B.S. Skinner, with the help of this rat Skinner box, elaborated the experiment of the effect of voluntary behavior on a consequence. So let's see. So what did Pavlov did? Before I go into the things, we should know what is a conditional stimulus and what is an unconditional stimulus. So a unconditional stimulus is something which is there, or unconditional stimulus and response is something which is there from birth. So here the unconditional stimulus is the food, and the response is the unconditional response is the salivation. So whenever the food is there in front of the dog salivates. So the food is the unconditional stimulus. It is there since birth, and salivation is also an unconditional response. Because of the unconditional stimulus, there is the unconditional response. Now, here, as we said, we are associating two stimulus. So we are going to associate a conditional stimulus with the unconditional stimulus. So what is a conditional stimulus? Conditional stimulus is a stimulus which actually does not elicit any type of response. But if it is repeatedly associated with the unconditional stimulus, then even in absence of this unconditional stimulus, we can see the effect. So let's see. This is the dog. The conditional reflex is ringing of the bell. So if the dog hears the ringing of the bell, nothing happens. There is no response. Now, if only unconditional stimulus like the food comes to the dog, obviously there is a response, and that is known as the unconditional response, where the dog salivates. Now, if repeatedly the conditional and the unconditional stimulus are given simultaneously, then we are also seeing the response, and that response is usually a unconditional, or the response to the unconditional stimulus. But after certain days, after this association remains for a longer period, this association of these two stimulus, when the unconditional stimulus is removed, with only the conditional stimulus, that is the ringing bell, the response is seen, and this is known as the conditional response. And this association with this experiment, Pavlov came to a conclusion and saw the cellular and mechanism formation that two stimuli, when associated for a longer time, even in the absence of the unconditional stimulus, the conditional stimulus can have a response. Now, as we said, operant learning was done by the Skinner's box. And we all know that in the Skinner box, there was a rat, and there is a liver. And the rat was inside. We had a electric. There was a wire which can induce electric shock to the rat, and similarly a place where a food can be dispensed. So the Skinner said that saw that if there is no response, suppose the rat touches the liver over here, but there is no response. So the touching of this liver was random. Whenever the rat felt like touched the liver. But if touching the liver, the food was given to the rat, that is the frequency of rat touching the liver was more. So that the pleasure, because the reward which the rat was getting as a food initiated the behavior change, which caused more number of times touching the liver. Now, if instead of when the rat instead of giving food, when the rat touched the liver, there was a shock given to the rat. It was seen that the behavior of the voluntary behavior of touching the liver decreased a lot. So a negative consequence decreased the voluntary behavior, and a positive consequence of giving food increased the voluntary behavior of the rat. So one more thing in classical conditioning, which is very important, was the timing. When the conditional response, the stimulus should be given, and when the unconditional stimulus should be given. Let's say a conditional, there is an unconditional stimulus, or we can say the food was given to the dog, but afterwards the bell was rung. Then there will be no effect. So the association to have a appropriate association between the conditional stimulus and the unconditional stimulus, it is very essential what is the timing of the stimulus given. So we can see over here when only the conditional stimulus is given, there is no response, the bell ringing. And here we can see that when the conditional response, when the conditional stimulus is given, the bell ringing, which is preceding the unconditional stimulus, that is the food. It is preceding the unconditional stimulus, and it also should overlap. There should be some overlapping with the unconditional. So the ringing should be overlapping it, and it should also precede it. Then only you see a response, and then only there will be established association, and later only the ringing bell can cause salivation or the response. And one more thing, while this association is being formed, if there is any other strong stimuli which is interfering, then the association between this conditional and unconditional stimulus will not be established. Now coming to the operant conditioning. Operant conditioning again has two types of behavioral changes, as we saw. One is, if increase in behavior, then we say it is the change of behavior is increased, then we say it is reinforcement. If there is a decrease in behavior, then we say it is punishment. Again, reinforcement can be a positive reinforcement or negative reinforcement, and punishment can be positive punishment and negative punishment. So we'll understand with examples. So positive reinforcement means, if a behavior, the kid has done the homework and the parent is giving something positive, so adding some positive stimulus to the already positive behavior what the kid has is called positive reinforcement. The other one is negative reinforcement, is the kid is keeping all the toys in place to avoid scolding from the mother. So to avoid the negative stimulus, the behavior is the change in behavior is seen. So this is known as negative reinforcement. Now coming to punishment, I'll give a better example which is there. So what happens in punishment? So there is decrease in behavior. We want to weaken the behavior, but we add a aversive stimulus or a negative stimulus. So how it is? Let's say a child is driving and he breaks, or he or she breaks the traffic rule. The traffic police gives a ticket that you have, or challan, to the individual. So that is to decrease the behavior. There was a aversive stimulus given by the traffic police. Now that same person, when goes home, the child goes home and tells parents that I was driving, I got a ticket. The parents said that for you, we are going to withdraw your driving, or we are going to withdraw your bike for a few days because you broke the rule. So there, what we are doing is we are removing the pleasant stimulus, the luxury or the pleasant stimulus, and making it. So this is known as a negative punishment. Now coming to the cellular mechanism of conditioned reflex. So already I have a little bit explained, but I'll just go to it. So as I said, This was also seen in aplasia. So what happens here? This is the sensory neuron, and this is the motor neuron to the effector organ, and where we see the response. So when only the conditional stimulus is there, there is no effect actually. But when there is a unconditional stimulus which is reinforced along with the culture, along with the conditioned stimulus, then there is through a facilitatory neuron, it is presynaptically and postsynaptically facilitating a response. So, and this is known as activity dependence. That means the conditioned stimulus is dependent on the activity of the unconditioned stimulus to be associated, so that in later, whenever, even if the unconditioned stimulus is not there, there is a response seen, and which is the conditional response. So this was, again, as I said, it was studied in the aplasia, and here this one is the unconditional reflex, and this is the conditional reflex, because normally when we stimulate the tail, this is innate, we see the gill withdrawal, but here the mantle, if we just stimulate it, there will be no gill withdrawal. So this is a conditional reflex. But if these two reflexes, if these two stimuluses are associated for a longer period, it has been seen that there is both presynaptically and postsynaptically facilitation of neurotransmitters, and or facilitation causes the response of the or gill withdrawal is seen more. So this is the molecular explanation of the conditional and the unconditional stimulus, how it is actually increasing or facilitating. So as I said, the unconditional stimulus, the facilitatory neuron, is affecting both presynaptically and postsynaptically, but the conditional stimulus is only affecting presynaptically. So how does the unconditional stimulus stimulate? It does by the 5-HT pathway. We have already said how the cyclic AMP causes activation of the PKA, and it modulates neurotransmitter release. Now, the conditional stimulus also activates the calcium channel, opening of the calcium channels, causing more calcium influx. And this calcium also activates the PKA activating system, or modulates the PKA activating system, and also directly causes release of neurotransmitter release at the neuromuscular, at the synapse. Now, the unconditional stimulus also acts upon the motor neuron, and it depolarizes the motor neuron, and the neurotransmitter release, glutamate released, comes and attaches to the NMDA receptors and causes influx of calcium. The unconditional reflex, depolarization of the motor neuron through the unconditional stimulus, and the calcium influx, both has a facilitatory effect on the motor neuron. And this calcium influx also gives a retrograde signal to the presynaptic or sensory neuron to enhance all these activity. So the neurotransmitter release over here is also facilitated. What I'm trying to say is this becomes a this retrograde signaling, or the modulation of conditional stimulus causing glutamate release and acting on the NMDA, when paired with the depolarization of unconditional stimulus at the motor neuron, causes influx of calcium, and that calcium influx sends a retrograde signal to the presynaptic neuron to further enhance the release of neurotransmitters. So this becomes one way of facilitation, or a reverberating circuit where more amount of neurotransmitter is released. So in absence of unconditioned stimulus also, this phenomenon works, and that is how the association takes place. So the neural mechanism of operant conditioning is also similar to the classical conditioning at molecular and the cellular level. Now, if you see the physiological basis of the conditioned reflexes, there is formation of, because of repeated association, there is formation of new functional connections in the nervous tissue. So here, in the case of Pavlov, the auditory, the ringing of the bell, there is some auditory pathway connection with the autonomic connection, but it's not a direct one. So there is functional new connection made, and it has been seen that the site of new connection can be either intracortical or subcortical. And how was this how did they come into this conclusion? Because they, when in experimental animal, in a decorticate animal, they saw that conditioned reflex is not building up. So that means the cortex is one side of conditional reflex. Now when they removed the whole neocortex removed the whole neocortex, they also saw that some amount of non-discriminative conditioned reflex is happening. What is a non-discriminative conditioned reflex? Here, the non-discriminative conditioned reflex means if the organism is able to only identify the stimulus which was only associated with the unconditional one. But if there is a new stimulus, then the organism is not able to identify. So the non-discriminatory conditioned reflex was even in the absence of neocortex was built up. So they thought that some amount of at subcortical level also there is new functional connection taking place. Now, the third learning theory, which has not been elaborately is one is observational theory, or the observational learning, or social learning theory. So this is studied by Albert Bandura, and what does it say? The individual learn new behavior through observing or imitating others. So here, Isla is watching Kim's go near a candle, and see that Kim is burning his finger. So now, and also he sees that the Kim is having pain in the finger because of the burning. Now, Isla avoids going to the candle because he has seen that the behavior which was done by Kim is giving a negative consequence. So there are certain steps in which this theory learns, and we see that this we can see also in human beings. When the kid is growing up, the individual observes a particular action, pays attention to see what has happened to what is the consequence to that particular action, then remembers both what happened, what was the action and what was the consequence. And in later life, if the action was a good one or the consequence was a good one for the action, repeats it or reinforces it. But if it was a negative one, then avoids it. Now coming to memory. Memory can be classified according to the storage. According to duration of storage, memory is classified into sensory memory, short-term memory, intermediate memory, and long-term memory. Actually, I went through many articles. Many researchers differ in saying exactly what the classification of memory actually varies. So some say that it's short-term, intermediate, and long-term. Some say that it is sensory memory, short-term memory, and sensory and short-term memory is one type of working memory, and there is no intermediate memory. It is directly long-term memory. So I thought I'll just keep everything and just explain a little bit about it. So what is a sensory memory? Sensory memory is when we anything new we see. Let's say I'm sitting in a class and I'm seeing a PPT, something new I heard or I saw something new. So that is the sensory memory. So I immediately receive that new thing into one. And after some times, obviously that it doesn't stay in the memory, it goes off. And sensory memory can be of different types. As if we see something and learn, or remember, that is iconic memory. If we hear something and learn, then it is echoic memory. And if we feel something and learn, then it is haptoic memory. And these memory, basically, when we see, and here we can say this is something known as a priming time. So our brain is being primed to something new, and so that information is first time going into the brain. And these usually go and they go to the cortex, but they don't get stored, because it's for a few for seconds. After that, we forget about it. Now, what is short term or recent memory, or primary or active memory? So short term memory is also known as recent memory, primary memory, or active memory. So it lasts for seconds to minutes. Some say it is a one type of working memory, but many differ about it. And see, so what is example what of short term memory is, like something which is happening recently, what I ate today in the breakfast, or what I read in the morning, what some my husband said in last night. So maybe even again, there is a confusion regarding the duration, because some say it is like minutes to days, and intermediate is days to week. So there is a confusion. So what to short term memory is a memory which stays for a time beyond which we forget it. And short term memory is always a the capacity of short term memory is limited. So if something new comes, we forget the previous one. Now, next is intermediate or secondary memory, and long term or remote or tertiary memory. As I said, intermediate and secondary memory, again, when I explain the different stages of memory, or mechanism, how these are stored and all these things, then I'll explain where it is. And long term memory is something which we remember for a long time, but again, there are certain researchers who say that anything beyond 30 seconds, if we remember, it is long term memory. I'm not sure of this, but there are researchers who are saying this. So the person you met in the coffee shop, that in last week, that is also a long term memory. And remembering your friend's birthday is your long term memory, and obviously remembering your Aadhaar card address is also your long term memory. Now, coming to classification of memory, because as I said, these memories are not permanent. So if we see the long term memory are of two forms. One is explicit memory or declarative memory. The other one is known as the implicit memory or the non-declarative memory. The implicit memory is something which is known as reflex learning. So what happens basically, or we are not aware of the implicit memory, but we are aware of the explicit memory. Here, this memory comes involuntary, but here we voluntarily recall the explicit memory. Now, I'll give one example. There are different types of implicit memory again. There is priming, there is procedural memory, associative learning, and classical and both operant and non-associative learning are also memory, or through this in implicit memory is more pronounced. So we'll see examples of this. So priming, as I said, we hear or see or read something very new. So that is implicit memory. We skills or habits which we have acquired by repeated by doing it repeatedly, that becomes a procedural memory. Like example is driving. Cooking, we don't remember I have to put salt, now I have to put water, I have to put vegetables. By repeatedly doing the things, it has become a procedure, we know it. Similarly, we while driving, we don't think that I have to put the clutch now, I have to put the brake now. So by repeatedly doing the thing, now it becomes a procedural thing and it is involuntary. And as I said, associative and non-associative. This also we are not doing it we are not doing it voluntarily, but it is coming. So suppose there is a bad event associated with something, so we remember that for a long time. So that becomes, that stimulus is associative, the bad and good one. So here it is an emotional response. Similarly, we have response, as we have seen in the dog. One stimulus is causing a response in the uh, muscle or salivary, this is a motor response, then it is, we said, is skeletal muscle. So here we can see the different involvement of different parts of the brain also. Similarly, non-associative learnings are also reflex pathways. And here we can see the priming is, as I said, it goes to the neocortex. Now, remember, when I explained the sensory motor, I said that the individual learns, the priming is through the learns and by seeing, by feeling, and by hearing. But once that becomes a long-term memory, then we know it goes and gets stored in different parts of the neocortex, either the uh, uh, occipital association area, the parietal association area, or the your visuospatial association area. So that is where it goes and it gets stored. Now, if you see the procedural ones usually are, are there, or the involvement of stratum is there. And for emotional one, it is amygdala, and for skeletal musculature, it is cerebellum. And for non-associative learning, all the reflex pathways, including the sensory and the motor one. Now, if we see the explicitive one, explicit memory or the declarative memory, it is something which is voluntary, or we are aware of it, we think of it. Again, it is of two types. One is semantic and the other one is episodic. So semantic is factual memory and episodic is event memory, or memory which is experience, which is there through experience. And here the memory is involving both hippocampus and medial temporal lobe. So we'll just go through the memories one by one. So if we just differentiate the implicit and the explicit memory, also known as implicit memory is also known as non-declarative memory or procedural memory, and this is known as declarative memory. Now, conscious awareness is not there in implicit one, but here it is conscious awareness there. We are thinking, we are remembering, yes, I went to my friend's marriage 15 days back, and I met an old friend of mine. So this is explicit memory. Now the task involvement over here is automatic performance without any conscious effort driving, and here involves conscious retrieval or recall. So I am remembering I went to the friend's marriage. There I met the friend who was studying with me in the school. So these things are recall is there and conscious retrieval is there. Now here, if we see the types, we have the priming or skill learning and habit forming. All these things are there. Conditioning and non-associative conditioning is there. And here the types are episodic or semantic. Now flexibility, this is less flexible, but this is highly flexible. What we understand by less flexible and highly flexible? See, whatever, if the dog has the bell was the ringing bell was associated with the food, then the response was seen. So here if something else, now suppose we whistle, if we whistle, there will be no response because only the association was seen between the ringing bell and the food. So this type of memory is usually less flexible because whatever initially learned, that is how it is, that is how it responds. So we cannot modify it. But here it is highly flexible. The information is linked to context. So how it is, suppose the individual is remembering, I met the friend in the thing and that friend was studying with me this time, then suddenly somebody else enters in, and at that time someone else enters, that the recalling of that thing is no more there. Something else comes. So this is highly flexible, but this is less flexible. Now, learning conditions, as I said, these are very close to the original learning conditions, but here associated things also can happen. So maybe from my recall, I'm remembering that I should not have said this to my friend, but then suddenly I realized, no, that the way that friend treated his father was wrong. So okay, it's okay, I have done it in the correct way. So what I'm saying is, in explicit type of memory, other factors are influencing it. So the response may be a little bit different. So examples, as I said, driving, cooking, swimming, all these are because of the implicit memory. And here, going in a vacation or going to any friend's house and recalling the events, anything, searching your keys, thinking where I kept my keys, all these are explicit memory. Now, coming to, actually, I've already explained what is a semantic memory. As I said, explicit memory is of two types. One is semantic, and the other one is autobiographical. Here, the semantic one, or the factual knowledge, which tells us about any facts, objects, or concepts. So we know the capital of India is Delhi. We know the capital of Tamil Nadu is Chennai. So all these are facts. So the memory which is there is, these are known as semantic memory. And we know the definitions of the meaning of the words. We know, so it becomes as I said, these are part of the long-term memory. So gradually priming the facts makes us remember for a long-term way, and this type of memory is known as the semantic memory. And there are different places, these neocortical areas, at different places of neocortical areas, the different types of memory, different types of memory in according to origin is stored. Like, suppose we remember a smart key of a car. What do we think of? We think that if somebody tells about the smart key of the car, we think of a small structure, what we have seen, and what is the color of the structure. It has keys, and each keys has unlock and lock button. Similarly, we think that it sounds like this. Similarly, we think that it is made up of plastic or metal. How does it feel? So what I'm trying to say is, when we recalling this, it is coming from different neocortical areas, and we know that is the occipital association cortex, auditory association cortex, or the parietal association cortex. So, and it has been confirmed that if there is damage to specific cortical areas, there are different types of symptoms seen. So one such symptom is known as the associative visual agnosia, where if there is a lesion to the posterior parietal cortex, the individual cannot name the object, but can tick upon the diagram. The individual, by seeing the diagram of the object, can say this is the object. So here, cannot name it, but can draw it, or can show the right drawing of that object. Similarly, we have appreciative visual agnosia, where the occipital association cortex is, there is a lesion. So here, the individual cannot draw the object, but can name it, or name it, that too with some amount of cue, or with some amount of direction. The person can name it. Now, what is the episodic memory or autobiographical memory? We already said, we have already explained. This is the memory linked to personal event or experiences, and it is usually stored in the prefrontal cortex, and then it goes to also, and it is linked also with the different association cortex. Now, that's why the frontal lobe damage leads to source amnesia. Source amnesia means from where the person learned it, that thing he or she cannot remember. Now, coming to the mechanism of implicit memory. So there are short-term storage mechanism and long-term storage mechanism. In short term, we see that basically short-term storage mechanism is through effectiveness of synaptic transmission. Either synaptic transmission is increased or decreased. So that is through a habituation or sensitization, and through associative learning also. And we already know that in habituation there is decrease in neurotransmitter at the presynaptic level, and sensitization there is increase in presynaptic facilitation. And in associative learning, both presynaptic and postsynaptic facilitation or inhibition is done. Now, if we see the long-term storage mechanism of implicit memory, whenever in long-term, we understand about long-term memory, there actually the short-term memory enters into a stable long-term memory, and that the process is known as consolidation. So here we will go in details while I explain the mechanism of explicit memory. What is consolidation? So what are the processes involved in consolidation? Gene expression and new protein synthesis and growth of synaptic connections. And already we have seen that how gene expression and new protein synthesis takes place through the Krebs signaling pathway, while I explained the long-term effect of sensitization. Now, coming to mechanism of explicit memory or declarative memory, basically there are 4 stages of processing memory, or processing of memory. One is known as encoding. Next is consolidation, storage, and retrieval. If one of them is not happening, then we cannot say it is long-term memory. And encoding means initially, when we initially receive any information, how it enters or how it enters to the brain and how it is processed is known as encoding. Now, next is consolidation. Once the process of encoding finishes, how it consolidates from short term to long term, and what are the things which is done. Now, once consolidation is finished, where it is stored, and then how it is retrieved. So these are the 4 processes of long term or long lasting memory. Now, coming to encoding, as I said, encoding is always it is a process where awareness or attention is required. Without awareness and attention, it is very difficult to encode the things. In implicit memory also, initially whenever we are, implicit memory also it's occurring reflexly, but still then initially when someone is starting to cook, someone is starting to drive, or someone is starting a swimming, initially there is some amount of awareness or attention there. Because of repeated behavior, that it becomes an implicit memory. Otherwise, initial learning is, or initial part of learning remains, and that is through awareness and attention. And what happens next is encoding depends upon the retention in hippocampus and other parts of the medial temporal lobe. So basically, it's the process where hippocampus and other parts of the medial temporal lobe plays a role. We'll see what are the places. So whenever we learn something, we know that through our visual association, auditory and palatal or sensory association area. From there, the information in series goes to the parahippocampal cortex and perirhinal cortex, and from there it goes to the anterorhinal cortex, and from anterorhinal cortex it goes to dentate gyrus, different parts of the hippocampus, and subiculum. And from these areas again, all the information comes back to the anterorhinal, the parahippocampal, perirhinal, and again to the different parts of the different parts of association neocortex. So what basically occurs is, so in the process of encoding, there is continuous reverberating circuits which work between the neocortex to the hippocampal area and associated area, and again back to the neocortical areas. And this causes either a long-term potentiation or inhibition. And along with that, there are many molecular levels where accumulation of calcium to effectively to increase the efficiency of synaptic transmission, there is accumulation of calcium in the axon terminals. So the main thing is, while encoding, there is repetitive or continuous reverberating circuit working between the different different association neocortical areas to the hippocampus and the medial temporal lobe, and again between these two these structures, and that is known as encoding. Now let's see what do you mean by a continuous reverberating circuit. Actually, this was established by Donald Hebb, and it is known as the Hebb's rule. What does Hebb say? What did Hebb say? Hebb said that when the axon of a neuron is stimulating the B is also stimulating the A. So, and in this process, A is stimulating B and B is stimulating A. In this process, what is happening? There is the persistent firing of both the neurons causes some growth process and metabolic change that takes place in both the cells, and that only leads to next step of memory. Now here, with example of two neurons, we have shown the continuous reverberating circuit, but actually if you see there is continuous, these circuits can be polysynaptic and very complex reverbation takes place. Now coming to the long-term potentiation and long-term depression, which also helps in change in the synaptic strength. So increase or decrease in synaptic strength depends upon the long-term potentiation and the long-term depression. So here you can see in long-term potentiation there is an increase in synaptic efficiency, but in depression there is a decrease in synaptic efficiency. This is due to high frequency stimulation, and depression is due to low frequency stimulation. And here what happens, what is the mechanism? At molecular level, there is increase in AMPA or NMDA receptors, which enhance the synaptic strength post-synaptically. But here there is decreased AMPA receptors, AMPA or NMDA receptors, which leads to a decreased synaptic strength. And if we see the brain region where it is most studied, the hippocampus is studied for crucial for spatial and associative learning. So long-term potentiation, as I already told you, it is more seen in between the neocortex and the hippocampus, and depression is seen in cerebellum. Depression is seen in cerebellum for fine-tuning the motor skilled motor movements. And this is usually long lasting. Both of them are long lasting. And calcium influx, as I already said, calcium influx is causes this strengthening of the postsynaptic one. But here, along with calcium, the other signaling methods are also. Now coming to the next step, that is consolidation of long-term memory. Again, hippocampus again plays a very important role in long-term memory in the process of consolidation. So what happens once the encoding has taken place? Now there will be consolidation taking place, so that only if the process of consolidation occurs, then only we can say it is a long-term memory. In short-term memory, we have some amount of encoding taking place. We have encoding taking place, but for long-term memory, if consolidation does not take place, we cannot say that encoded information has become a long-term memory. So what are the physiological processes involved? Again, there are two types of process. One is known as rehearsal mechanism, but this is actually too simple to explain this. It is very complex, but I have just made it very simple. One is known as a rehearsal mechanism, and the other one is known as a synaptic plasticity. So the rehearsal mechanism is, as I already said, through the reverberating circuits again, the expression of genes, synthesis of new proteins, and also there is synaptic structural changes in at the synapse, where there is available of more number of synaptic vesicles having the neurotransmitter, the releasing site of these vesicles also increase, and the synaptic terminals also increase, and also there is a change seen in the postsynaptic spine. There's also change of shape seen in the postsynaptic spine. Now, synaptic plasticity, how does it work? Basically, synaptic plasticity is the brain's ability to modify the strength of connections between the neurons over time. So, and it is usually in response to experience. So, with experience, the adaptability of the synapse is known as synaptic plasticity. And again, the synaptic plasticity depends upon long-term potentiation and short-term, long-term depression, and etc. And it has been seen that when emotion is related to some information, it enhances consolidation. And what is the evidence? And we know that the site of emotion is amygdala. And what is the evidence? If there is a lesion to the hippocampus and amygdala together, the loss of memory is more if it is only to the with respect to only listen to the hippocampus. That's why it has been seen that any information, if associated with emotion, is better, or is consolidated in a facilitated way. Now, what are the different sites of memory storage? Actually, we have discussed this. So, in the past part also I said. So, the sensory motor memory, if we see short term one, it goes to the different parts of the neocortex, but it does not stay for a long time. Now, working memory, I just tell what is a working memory. I think I forgot to exchange the slide. So, working memory, here what happens? The auditory and visuospatial one, it stays in the prefrontal lobe or the frontal lobe. Coming to the long term, we have seen the explicit memory, the episodic one and semantic both, they are in the hippocampus and the medial temporal lobe. And the implicit, as we said, it is a different side depending upon if it is procedural skill, then it is striatum. If it is some response, a motor response, then it is cerebellum. If it is emotion, then it is amygdala. And all the classical and the association between stimuli and behavior is through the habituation, sensitization, and the classical and operant conditioning through the reflex pathways. Before I explain this, I'll just go to the working memory. So, what is a working memory? When the process of initial coding has taken place, and then we're recalling it. So, actually, when we see after that, we have a step that is known as a retrieval of memory. So, how retrieval of memory takes place? Retrieval of memory takes place in we voluntarily recall the thing, and it is not it from the different sites, from different sites where the things are stored, the information is collected, and we recall that. And and when do we say it is a working memory? If encoding is there and then recalling is there, but there is no process of consolidation, then we say it is a working memory. So basically, any work which has to be done, till that work is over, the memory which is activated is known as working memory. Once the work is done, we don't remember the fact. Let's say opening a bank account. So we remember these are the documents required. We have to fill the form. All these things we have it in our mind. And after it is done, after many days, if somebody asks how to open the account, we say I don't remember, I just submitted such certain documents. But at that time, when we are doing the task, we were aware of all the things. But now we are not aware of the things because we forgot it. Because once the working memory is, once the task is over, we don't retain the memory no longer. So it is one type of a short-term memory actually, and the process is attentional allocation system. As I said, it involves the prefrontal cortex or the frontal lobe. And how do we do it? It is by a rehearsal system, by the articulatory loop, or by the visual-spatial sketchpad. So articulatory loop means we remember, okay, I have to, so we articulate subconsciously or even sometimes loud. Okay, Aadhaar card, I need the Aadhaar card, I need the PAN card, I need this, I need the telephone number, I need a Aadhaar, which is, so this is how articulatory thing we repeat through the articulatory method. Similarly, visual specialists, we try to remember. See, when we read a book and we are recalling, we try to remember, okay, I read about working memory, what I read about working memory. I try to remember what I read about working. So that is visual special sketchpad. So this is how the working memory here works, and usually it does not involve consolidation, so it never goes into a long-term memory unless and until if we repeat it and make sure that consolidation process takes place. Just one thing I forgot to say during the consolidation, when the information from encoding to consolidation occurs, it occurs in bits of pieces. How does it occur? The brain, the hippocampus, and the medial temporal lobe, these they compartmentalize the information. They first see that what is the information, what is already known, what the brain already knows, and then what new information the brain got, and it works upon the information, the new information what the brain actually got. So that is how it works. So gradually, once one information is consolidated, we remember it for a longer period. Anything new coming into that knowledge, if repeatedly it is encoded, then it becomes a consolidation and stays for a longer period. Now, what happens if there is failure of consolidation? Before we go to the failure of consolidation, we'll see what happens to the memory transfer. Is there any hemispheric or memory transfer? So basically, there was a experimental which was conducted. Here we can see in the first picture, individual is told about something. The individual is given a visual task to discriminate a visual task. Now, and the left eye is closed. Now, when the right eye closes, the right eye is closed where the individual learned it, and the left eye is told to identify the problem. Now, the individual can identify the problem because the learning which occurred, it passed through the optic chiasma and also the corpus callosum, and both the hemisphere got the information. So here the left eye, through the left eye also could be able to identify the problem. So this is telling us that both the optic chiasma and the corpus callosum are helping in the thing. But then they saw that, let's see what is happening. Is it only both these two things, or singly also it can happen? So what happened is, before training the individual, the optic chiasma was dissected, and now they trained the right eye with the left eye closed, and then the test eye was the left eye. Still then the individual could able to differentiate the visual tasking or identify the visual tasking. That means even in absence of optic chiasma, the corpus callosum is helping in the interhemispheric transfer of the learning. Now, next what they did is, before giving the training to the right eye, they cut they split or they dissected the optic chiasma. They also dissected the corpus callosum, and then they made the right eye to train the visual task. Now, when they tested on the left eye, the individual or the left eye could not differentiate or could not identify the problem in the visual discrimination task. So this indicates that because there was no optic chiasma and corpus callosum, that is why there was no interhemispheric change of information from the right hemisphere to the left one. And what basically happens? Not only optic chiasma and corpus callosum is responsible together, individually also they can cause uh, interhemispheric memory transfer. And one more thing I just want to add up over here: the anterior aspect of the corpus callosum usually carries uh, uh, carries um, the posterior aspect carries the visual memory. The anterior aspect carries the your spatial visual spatial the auditory and the spatial memory through the commissural fibers. Coming to the effect of failure of consolidation, this is actually what happens in Alzheimer's, where there is a uh, there is a defect in the hippocampus, or even the bilateral destruction of hippocampus and medial dorsal a temporal lobe also can cause this. And what happens is there can be anterograde amnesia or retrograde amnesia. So we know that amnesia is inability to remember things, and here anterograde amnesia is inability to establish new long-term memories. So new things we cannot learn. And in retrograde amnesia, the past memory we usually also is lost, but the remote ones are there, because in the individual, the consolidation, when the consolidation is lost, the things which were not consolidated are only lost. Not the things which were consolidated previously, very previously, they are not lost. So something from the childhood or from remote is there, it will exist, but something just before the injury to the parts will there will be loss of memory because of absence of consolidation. Now, the Wernicke's Korsakoff psychosis, it is a condition seen in chronic alcoholism. There is basically thiamine deficiency, but there can be other causes like malnutrition also. Here also we find there is retrograde amnesia and anterograde amnesia, and there are two stages. One is Wernicke's encephalopathy, where the individual is in a confusional state, and Korsakoff syndrome, where there's a long-term memory impairment, and because of that, there is neurological problems. Alzheimer's disease, I'll not go into that details. I think this is a neurodegenerative disease, and which occurs in elderly patients, and that leads to dementia. And basically, the neocortex, the entorhinal area, hippocampus, amygdala, these are the areas which are affected. We know that neurofibrillatory plaques, Lewy body, and different angles, these are formed. And senile dementia is again dementia which is seen in elderly people, and one of the causes is Alzheimer's disease. But along with that, we have vascular dementia, and there are different causes of senile dementia. And here also there is impairment of memory and all the cognitive capacities. So thank you. I think I tried to put it as easy it could be. And this is my institute basically. The left one is Kalinga Institute of Medical Sciences, where the medical college in our university where I work. And this is actually, it's a very big, our university is a very, it's there in a over a very big area. And this is the main site, KIT University, and it's a very beautiful university. I welcome all of you. Please do visit, and if you are coming, please do connect to me. If you wish to visit, also do connect to me. We will really appreciate. I have called Priscilla ma'am so many times. She had come once because there was a pulmonary national conference on pulmonologist, I think. So she had come. I request Muthu Pratibha, I request everyone actually basically to come once and visit our university. Thank you. Please, any other things, any doubt or anything for discussion? It's open for discussion.
Thank you, Magna.
Should I stop sharing? Should I stop sharing the PPT?
Thank you, Magna, for that enlightening lecture on a very complex topic. Now the forum is open for question and answer. You can raise your queries to our guest speaker now. Please unmute yourself and ask questions if you have any.
Okay, Avik, shall I, there are people whom I can see have raised their hands.
Yes.
Okay.
Yeah, please unmute yourself and you can raise your queries to our guest speaker. Please
Mr. Mohammed
speak up.
Yahoo, can you please?
Yes. At least post the chat box. We'll take up your questions.
PS. There was one more hand raising. PS.
There are no questions in the chat box as
When
well.
When there are no questions, there are two conclusions. One is the speaker was not good, nothing could be understood, or the speaker was really great, everything understood.
Really great. It was really great, Magna. Yeah, sure. That's for sure. Yeah, you can put it up in the chat box. Now, let's take a group photograph. Now, I will hand over the virtual stage to our Master of Ceremony, Ms. Nachia.
Dr. Magna ma'am, your insight truly rewired our understanding of how memory works, and your session was indeed a neural symphony. I'm sure what we have learned today will be consolidated into a long-term memory.
It should.
Thank you.
But for that, you have to go through it.
As we have come to end of this engaging session, we would like to commemorate the occasion with a group photo. Kindly request all the participants to turn on their camera for a few seconds so that we can capture the special moment together. I request the participants to turn on the cameras. Thank you all. I request all the participants to fill the post-test forms and feedback forms. The link will be shared in the chat box. Now, I would like to invite Dr. Suganya ma'am, Assistant Professor of Physiology, to deliver her vote of thanks.
Good morning, all and all. It's indeed a great pleasure for me to extend the vote of thanks to this memorable occasion. First and foremost, I would like to express my sincere gratitude to the esteemed members of our management, our Honorable Chancellor, Pro-Chancellor, Vice Chancellor and Pro Vice Chancellor for providing us with the essential infrastructure and for the constant support in all our academic events. We are deeply thankful to Dr. Balaji Singh Sir, Dean Medical College, for being an unwavering source of encouragement and support in all our academic activities. Our heartfelt thanks to our distinguished guest speaker, Dr. Magna Ma'am, for generously taking time out of a busy schedule to share her knowledge and expertise with us. I would also like to extend a heartfelt thanks to our head of the department, Dr. Muthupradipa Ma'am, for organizing and facilitating such thought-provoking guest lectures that enhance learning among the postgraduates. We are grateful to our respected senior professors, Dr. Padmavati Ma'am and Dr. Prisla Johnson Ma'am, for their consistent guidance and encouragement. A special note of thanks to our professor, Dr. Abhirami Ma'am, coordinator of the PALS 4 team, for her dedicated efforts in planning and coordinating today's program. I also thank our postgraduates, Mr. Edgar for handling the audiovisual arrangements, Ms. Nakshia for our admirable anchoring of the event, Mr. Allen and Ms. Gayatri for taking care of the video recording and photography. I sincerely appreciate the contributions of all our faculty members, technicians, Secretary Ms. Batmakumari, and other support staff of the department for their hard work and commitment in making this successful event. Most importantly, we thank all the participants for being a part of this program. Thank you all once again.
Wow, after this session, I think now, as a token of our gratitude, we would like to present a certificate of appreciation to our esteemed speaker, Dr. Magna Ma'am, for taking the time to share her valuable insights with us today. Thank you so much, ma'am, for enlightening us with your lecture. To learn is to live twice, once in the moment and once in memory. Let's carry this learning forward, not just in journals or exams, but in how we think, teach, and grow. So let's keep this momentum going. Now, I request the participants to complete your post-test form and feedback form to receive your e-certificate. Join us for the next month for PALS 5, where learning continues and memory grows. Thank you all once again for your presence, attention, and energy. Until next time, keep your neurons curious, keep thinking, keep questioning, and keep remembering. Nandri, Dhanyavad.
Thank you.
Thank you, Magna. Thank you so much for joining us today.
Thank you. Avi, I'm leaving the call.